Okay, let's get started. I'll share my PowerPoint here. And here we go. Last time we talked about bone at the cellular slash molecular level. Bone undergoes a complex loading environment due to its complicated geometry, considerably more complicated than cartilage. And perhaps because of that complicated loading environment, the cellular organization of bone is more complicated than cartilage. Cartilage had just one type of cell. Bone has three types of cells: osteoblasts, osteoclasts, and osteocytes. The osteoblasts lay down new, healthy, hopefully stronger bone. The osteoclasts tear down older, damaged bone. And the osteocytes are responsible for the communication and organization amongst those cells to kind of coordinate that. Like you'll see at basically everything at the cellular slash molecular level, those cells are mechanosensitive, which means the way to trigger them and signal them to engage in that metabolic activity is not through a metabolic signal, not through a neural signal, not through a cardiovascular signal. It's through a mechanical signal. You deform the cells, changing their shape, and that triggers or initiates the signal in them to get them to do that job, to engage in that communication that performs that metabolic activity. So there's not the metabolic side and the mechanical side, and these are separate things. They're intimately tied together and depend critically on mechanical loading. So mechanical loading was not just important or good for the health of bone, it's essential for the health of bone. The way that bone repairs cracks at the microscopic level is kind of interesting. You have these cracks in bone, and the osteoclasts, what do they actually do to an osteoblast? Like, what do those cells actually do to repair that crack? The osteoclasts go in and literally like eat it. Like they just bite like a chunk out of that crack and just, replace the crack with a hole. And then the osteoblasts go in and fill in the hole. So you tear it down and then you fill it in, and that's the dual roles of those two distinct cells in cortical bone. Okay, moving on today, and we will talk about tendon at the cellular level and also ligament at the cellular level. This was a set of lectures that I added new fairly recently to the class, thinking that I'd spend like a whole lecture on the cellular level for tendon and a whole lecture on the cellular level for ligaments. Turns out tendons and ligaments are very similar at the cellular level. Like if you were looking at under an electron microscope, looking at like a strand of tendon versus a strand of ligament, you'd be hard pressed to tell distinctly beyond like a coin flip guess, like is that a tendon or is that a ligament? So they're quite similar at the cellular level. At the tissue level, of course, they are very distinctly different tissues. The tendon connects muscle fibers to the skeleton, and ligaments connect from bone to bone on the skeleton. So they play fairly substantially different roles at the macroscopic tissue level. But the microscopic cellular slash molecular level, tendon and ligament are quite similar. So we'll cover both of them today in this particular lecture. So what does tendon look like at the cellular slash molecular level? If you zoom in on it real close from the macro scale here on the left to the micro scale, you see these little strands of material making up tendon. Tendon, like most elastic stuff in the body, is primarily collagen. So those little strands that you're seeing are collagen. Notice that they're kind of wavy, right? They're kind of crimped a little bit. And notice that you see these little discolored, kind of darker purplish patches in this sea of like pink, crimpy collagen fibrils here. Those little discolored patches are cells, and specifically those are tenocytes. Teno, you might imagine, means tendon, and site, as we've learned, means cell. So it's a rather uncreatively named thing, tenocyte or tendon cell. So those are the cells in tendon that do conceptually many of the same roles that we've talked about cells doing in other tissues. They're responsible for regulating the health of that tissue through combining the metabolic activity of the tissue with the mechanical loading and mechanical activity of the tissue. So what does tendon look like at a micro scale? We'll first cover tendon And then we'll kind of generalize that to ligaments towards the end of the lecture. So what does tendon look like at the cellular slash molecular, microscopic scale here? Like much thing, or like many tissues, tendon has a solid phase and a liquid phase. That solid phase is the extracellular matrix, so the solid stuff extra or in addition to the cells, so kind of similar to cartilage there. Made up of about that solid phase makes up about 40% of the mass of a typical tendon. Up to about 85% of that 40% is collagen. So just like cartilage, there's a lot of collagen in it. Collagen is a really abundant tissue throughout the body. Most like springy, elastic, deformable things in the body are primarily collagen. That's kind of what collagen does. It deforms and behaves in an elastic fashion. Nearly all of that collagen, there's lots of different types of collagen. The different types are kind of beyond the scope of this biomechanics course. But nearly all of the collagen in tendon in particular is type 1 collagen, which is these long, kind of fibrous strands of collagen. Okay, so that's in total about 40% of the mass of tendon at the microscale. The other 60% is the liquid phase. And sometimes I'll screw up and call that water. It's not really water. There's a good chunk of it that is, cellular, molecularly H2O specifically, but there's a lot of other important stuff in it. So it's strictly speaking, it's synovial fluid. It's not water. Plays a similar role to cartilage in that it transports nutrients in and out of that tendon. And that's similar to the role that fluid played in cartilage. That's very important because many tendons, not all tendons, but many tendons are poorly vascularized, meaning they don't have much connection to the circulatory system. Unlike healthy cartilage, healthy tendon often does have direct connections to the nervous system. There's Golgi tendon organs that you might have heard of in anatomy and physiology, or maybe in some of your motor control classes, where there's little Golgi tendon organs inside tendons that sense the level of force or stress inside that tendon, and that's through connections to the nervous system. So tendon does have a direct connection to the nervous system. It doesn't often have a direct, or at least a strong direct connection to the circulatory system. So there's really no way of the body like directly sending nutrients to tendon to help it with metabolism and healing and repair from damage and injury and things like that. So the only way to cycle nutrient-rich fluid in and out of the tendon is again to mechanically load it, to deform it and squish the old stuff out, and then undeform, remove the force deforming it and let the new nutrient-rich fluid flow in. So a really important element of tendon there for regulating its health through metabolic activity, linked to the mechanical loading of that tendon. Now, what does tendon look like at this structural level from the macro scale down to the micro scale? You've probably seen figures like this for muscle, where we examine, the different physical scales of muscle from large scales down to very small scales. And here's a similar one for tendon. So this would be kind of the whole tendon that you would see. Like this is the Achilles tendon, for example, here. Inside the Achilles tendon are these endotendons, which are kind of bundles of fascicles of tendons. Okay. Inside those fascicles is where you see the fibers, and that's the crimpy pink stuff that we saw on the electron microscope. Inside fibers are bundles of fibrils, and this is the level where we would see the cells appearing. And then inside fibrils are these strands of tropocollagen. Okay, again, the solid phase of cartilage being made primarily of collagen. And here we're getting down to like really, really small physical scales, like the nanometer scale there. Now, notice here in tendon, we see this crimping here at this physical scale, at the fiber scale. Then we get down to the fibril scale, and it looks like that crimping's gone. Right now everything is kind of straight here. Okay, so we have this crimp waveform. And this is an important concept to think about for tendon in general, but also for just stuff in general that elongates or changes shape. This is probably a concept that you've never really thought of before. But when something like is loaded and stretches and gets longer in response to that load, and then when you remove that load and it unstretches and goes back to its original unloaded length, like what physically is actually happening there? Like how does that structure, whether it's a tendon or a muscle or a piece of fabric or whatever, like how does it, what's actually going on that causes it to get longer? Like mechanically, yeah, we can say it's the force that's causing it to get longer, but within the thing itself that's lengthening, like what actually is happening to allow it to lengthen and get longer? Like what's going on there? So we'll try to explain that, like what's actually happening when we load something like a tendon with a force and it gets longer, like what exactly is going on there? What exactly is lengthening inside that thing? Now, here you've seen these sort of images before, but let's review it here since it might have been a while. Here is the relationship between the length of a tendon on the x-axis and the force expressed on that tendon from the muscle fibers connecting to it on the y-axis. And like most things, to stretch a tendon to longer and longer lengths here, that requires applying larger and larger forces to it, right? So we see as we go to the right on the x-axis, we're also generally going up on the y-axis. But notice that that relationship, regardless of whether it's this curve from one particular paper in the solid line or this kind of subtly different but conceptually similar curve from a different model in the dashed line, in both cases, that's a non-linear relationship, meaning it doesn't just progressively get longer at the same rate as we stretch it with larger and larger forces. At low force levels, the tendon is fairly compliant, fairly unstiff, meaning it stretches a lot in response to small increases in force. Okay. Then as we get to kind of a like an inflection point here, kind of in the middle, then it kind of abruptly stiffens up a little bit. Okay. And then it keeps stretching as we apply forces to it, but we have to keep applying larger and larger forces to get it to stretch smaller and smaller amounts. Okay. So it seems like there's kind of, regardless of like which curve we use here, which one we want to say is correct. They're both correct. These are just two different studies fitting these curves to two different sets of data. But in both cases, tendon has kind of an initial like low stiffness region as the force that's being expressed on it goes from zero force up to a moderate force. And then it kind of transitions to a high stiffness region as we keep increasing that force up from a moderate force to a high force, then tendon kind of behaves in a stiffer fashion. So we might think that maybe there's kind of conceptually two different mechanisms going on there for how tendon is actually getting longer. If it initially behaves as a not very stiff material, and then as we stretch it further and further, it becomes a more stiff material. So why does tendon lengthen when force increases? Like what is at the molecular slash cellular level, like what's actually happening there? Like what's changing at that level? And what's actually happening at the micro scale? Okay, this is something I never really thought about that much until I started teaching this particular part of this particular course. Like when my sleeve on my hoodie like stretches here, like I grab it and I apply force to it and it stretches, right? Like it's getting longer, but like, conservation of mass and all that, like by applying a force to it and making it longer, like I'm not making like more of my shirt, like it's not changing the mass of the shirt, it's just changing the shape of it, right? So, but what's actually happening to like the fabric strands, like the polyester and cotton inside my arm of my shirt there, like what's actually happening to those constitutive strands that make up this shirt? Like when the whole thing gets longer, like what's happening to those? I'm not magically creating more of them or bigger ones. Like something is changing in response to that application of force, and what exactly is changing there? So here is what tendon fibrils, those small, molecular level, physical scale strands that make up tendon, here's what's actually happening to them when they're at rest and unloaded. So a tendon that's just at its rest length with no force applied to it. And what's happening then when you apply a force to it as it goes from being unloaded at rest to now being loaded with some at least moderate, small to moderate muscle forces. Okay? When the tendon is at rest, the fibrils are crimped. Okay, so that's those wavy strands of collagen fibrils that we saw in the pink electron microscope picture early on in the lecture, and then also those kind of crimpy strands at the fibril scale in the drawing of tendon at different physical scales a little bit later on. So when tendon is at rest, those fibrils are crimped, meaning they're kind of smushed like accordion style, right? So that's kind of their resting state. They have a crimp to them. Okay. As you apply larger and larger forces to those crimped fibrils. And what is that force? Again, this would be the force developed by muscle fibers and expressed on the tendon and stretching that tendon. Okay. So as you apply larger and larger forces from the muscle fibers, reflected in these figures in the size of this ball getting larger and larger and pulling those uh, tendon fibrils to longer and longer lengths. What's actually happening is the fibrils themselves aren't actually like elongating or stretching. What's actually happening is they're uncrimping. Okay, they're rotating from something like this at the top at rest, where they're very crimped. Those crimps unfold through rotation as forces rise. Okay, until you get to a relatively high force here, where they're now fully uncrimped or nearly fully uncrimped. Okay, so that's what's actually happening at the micro scale in a tissue like tendon when it goes from rest with no force applied to it up to a moderate force applied to it from muscle fibers. It starts in this heavily crimped state when it's unloaded. Okay. And then as you apply larger and larger forces to stretch it, those crimps uncrimp, allowing the entire thing to get a little bit longer. Okay. So that is what's happening here at this fiber slash fibril scale. Okay. Now, notice here we said that that's what's happening if I back up a few slides. That's what's happening to the lengthening of tendon in response to rising muscle forces from the fibers at these fairly low force levels and fairly short lengths here. Okay, so that's this kind of early low stiffness part of this tendon force extension curve. Okay, so that would stretch our tendon with about 2% strain up to forces that are maybe like, I don't know, 20 to 30-ish percent of our maximum isometric force. Okay. Those are fairly low strains and fairly low forces. To operate over like the full range of our ability to produce muscle forces, like for higher force tasks like sports and exercise and whatnot, we would load tendons with considerably larger forces, and it continues to stretch beyond that. But when we get to these kind of 2%-ish strain lengths here, that's the point where, if I go back here, we've already nearly fully uncrimped those tendon fibrils. Okay. So they've gone from being kind of crimped like this to being kind of fully uncrimped like that. Okay. And if I'm fully uncrimped like that, it would be a bad thing if I were to continue pulling and stretching those tendon fibrils. They're already fully uncrimped. And at that point, if I unload them, they're just going to go back to their crimped state. Okay. If I keep pulling on them beyond that, that's kind of like when you're pulling on a string and it's already like fully stretched at the largest length it's going to get to. And if you keep pulling on it, you're going to stretch it and permanently deform it and maybe fracture it and tear it. And that would be a bad thing, right? So we need some other mechanism here at higher force levels, going from moderate to high force levels through which tendon can continue to lengthen. And that's where this smaller kind of tropocollagen physical scale comes in. Okay, so what's happening as tendon lengthens from moderate force levels to higher force levels? Okay. Um, the best analogy that I can use for this is when I was a kid, we called these things Chinese finger traps. I don't know if they were actually Chinese in origin or not, but it was these kind of helical shaped structures that you would put your fingers in either side, and then if you pulled on them, they would kind of crimp down and pinch down on your fingers, and you couldn't get your fingers out, right? That's kind of what tendon looks like at the smallest triple collagen level physical scale here. It's these peptide chains that are kind of wound together in this helical fashion, okay, with these peptides kind of having some crimping to them, similar to what the tendon fibrils had at the larger physical scale, but they're crimped in kind of a rotational way rather than in a longitudinal way. So these tripocollagens exist inside these collagen fibrils. So here you're seeing those fully uncrimped collagen fibrils. And if I look at them at this point, when they're fully uncrimped at smaller physical scales, if I zoom in on one, when it's fully uncrimped and say, okay, what's it look like now at an even smaller physical scale? It will look like this up here, where those collagen fibrils comprise, or those tripocollagen fibers comprising these collagen fibrils are kind of crimped in a rotational way. Okay. Now, to lengthen further beyond this fully uncrimped state, I can still do that if I kind of unroll or unfold those twisted collagen fibrils. Okay. Now, here's what they actually look. These are, these drawings back here are just, cartoons and, estimates of what these things look like. Here's what those peptide chains in the triple collagen fibers actually look like for real. And you can see this isn't just like a conceptual idea. This is what they really look like. They're kind of twisted around each other along that longitudinal axis of those fibers. Now, you can see this kind of nicely here, how you could have two kind of distinct mechanisms of making this thing longer. Here it's still a little bit crimped. You can see this distinctly like wavy up and down pattern of this thing. Okay. And you can also see the peptide chains comprising the thing kind of wound and twisted around each other. Okay. So if I wanted to make this longer by stretching it, there's two motion-based mechanisms by which that could happen. I could undo the crimping, flatten the thing out, and that would make it longer. And I could also undo the twisting, okay, and that would make it longer. So that's those two separate mechanisms by which tendon at the microscale gets longer in responses to forces applied to it. Okay. At low force levels, like going from rest up to moderate forces, it's the uncrimping of the tendon fibrils. Okay. So they go from kind of accordion smashed to stretched out and flattened. And then at higher force levels, as I continue trying to stretch it further and further, beyond the point where those collagen fibers are fully uncrimped, then that's where the twisting or the untwisting of the peptide chain starts to happen. Now kind of like in exercise physiology, where we talk about like the three energy systems, and there's I'll just categorically distinguish between them. There's aerobic energy systems and anaerobic energy systems. It's not like you're using entirely one and then you get above some threshold level of effort and you completely stop using any of the aerobic pathway, and now we're using all of the anaerobic pathway. It's a gradual system where it's a gradual overlap where you're always using a little bit of all of the systems, and at some intensities you're using more of one and less of the other, but you're always using a little bit of both of them, right? Kind of a similar idea here for tendon. It's not like when I load it from low levels up to moderate levels, that's all from the uncrimping. And then at moderate levels to high levels, that's all from the untwisting. There's always a little bit of both going on, but at low to moderate levels, it's mostly from the uncrimping. And at moderate to high levels, it's mostly from the untwisting. And then as you get up to stretching it to literally its longest possible length that you could stretch it to without developing an injury, that's where both of those things would be maxed out. It would be fully uncrimped and fully untwisted. And any sort of additional stretch beyond that would be running a risk of like fracturing these peptide chains here, which would be an injury, or at least a mechanical definition of injury at the microscopic scale here. Yep. So not just a theory of Dr. Miller's here. These are established dominant mechanisms for how tendon actually stretches and recoils elastically at the microscopic scale. So two dominant mechanisms: collagen fibril elongation due to a largely elastic spring-like straightening of the tripocollagens, and then straightening of that fibril crimp that gradually increases over those successive loading cycles. Okay. So two separate mechanisms there: the uncrimping of the fibrils and the untwisting of the peptide chains comprising those fibrils at a smaller physical scale. And like I said, injury then would happen if you unfold or untwist those helices of the peptide chains that comprise the triple collagen, and then break the links or snap the links between those different collagen fibrils where the uncrimping largely happens. So that would be injury of a tendon at a molecular level that would then affect the macroscopic tissue level performance of that tendon and its ability to effectively transmit loads from the muscle fibers to the skeleton. Okay, haven't really talked much, despite this cellular level discussion of tendon today. Haven't really talked much about what the actual cells in tendons are doing. Tendon has kind of one type of cell, kind of not. There's tenocytes, and you can find various references that'll say, yeah, there's only one type of cell in tendon, it's tenocytes. But there's kind of several different like subtypes of tenocytes. Again, this kind of gets outside the scope of this, biomechanics class and the expertise of me as a non-cellular, non-molecular level expert. But there's kind of several different like subtypes of tenocytes that kind of get more involved in certain specific tasks and regulating like the health and the structure and the metabolic activity of tendon. But they're all just kind of different flavors of tenocytes. So it's not like bone where there's these distinctly separate cells that play distinct roles that don't overlap at all with what any other cell is doing. Tenocytes, there's several different subtypes of them, but they're all kind of broadly doing the same thing in terms of trying to maintain that extracellular matrix with the collagen fibers that give this thing its whole general structure and have these capacities for the uncrimping and the unfolding. So tenocytes, regardless of which subtype they are the cells of tendons. They regulate the metabolic activity of the extracellular matrix, which is again comprised primarily of collagen. And like cells in bones and cells in muscles, they are mechanosensitive. And you can see that here in the shape of these little purple tenocytes here, where they're kind of elongated. Oh, sorry, where they're kind of elongated, right? Meaning as you stretch this extracellular matrix through the cells being embedded in that matrix, that's also going to stretch those cells. And that stretch is a good thing. We talk about strain being like a predictor of injury. It's more, you kind of, you don't want to really think of that as like strain is always bad. Too much strain is bad. If you strain something too much, then that can cause it to be physically ruptured, and that's a mechanical definition of injury. But strain within like the normal ranges of strain that tissues are intended to undergo is generally a good thing. And at the molecular level, it's a good thing because that deforms those cells, and that's often the trigger for them to engage in their role in metabolism to regulate the health of that tissue. So that's what we mean by mechanosensitive. And then the cells in tendons are like that, just like the cells in bones were like that. Okay. Now, that activity of the tenocytes is, again, very important because tendons have a pretty poor blood supply. They're not like cartilage, where in good health they have literally no direct connection to the circulatory system. But some tendons have more blood supply than others, and some have very, very poor blood supply. So the regulation of the amount of like nutrition inside tendons that can help them repair and recover from even like normal levels of damage and especially like injury levels of damage is very important to actually load and deform the tendon to get it to trigger that metabolism. It's the only way to get it to trigger it in tendons that have a poor or no blood supply, which is quite common in some tendons. Now, during the initial stage of a tendon injury, which is typically called like the inflammatory stage, where it's like big and puffy and there's all sorts of fluid inside of it. During that stage, you typically want to immobilize the tendon. Okay. At that phase in tendon injury, like very early on after you've suffered like a tendon injury, stretching the tendon and loading it is generally bad at that stage. That's the stage where it's inflamed. There's a high level of kind of inflammatory activity and kind of immune system activity, trying to take care of some issues with the injury that are not heavily predicated on mechanically deforming the tissue. Okay, so early on in the stage of a tendon, especially when it's like kind of inflamed and kind of poofy, you don't want to load it at that point. However, after that initial stage subsides, which is usually a fairly short amount of time after a serious tendon injury, like usually within like a couple days or so, at that point, even when it might be a little bit painful, it's good to load the tendon. Okay, the pendulum has really swung here on recovery from injury due to some scientific discoveries on the role of mechanical loading in regulating the health and the function of tendon at the cellular/molecular level. The kind of clinical practice used to be, oh, you've got an injury, like it's got to rest, it's got to recover. So rest, 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 rest. Don't load it, don't load it, don't load it. Now that's changed quite a bit, where that's still the advice early on in this inflammation-driven stage. But after that inflammation has subsided, it's not just a useful thing, but many would say an essential thing, a highly beneficial thing at least, to actually load an injured tendon while it's in the act of recovery. Now, you don't want to do loads in terms of like, one rep maxes, like really heavy loads that could potentially injure it further. But, low levels of kind of moderate cyclical loading at that point are good for the recovery and repair of a tendon after an injury. That mechanical loading, again, because it's triggering the tenocytes to engage in their metabolism, increases collagen synthesis. And particularly what's been shown to be beneficial here, and this is a pretty prominent thing in physical therapy right now, is performing long, steady isometric contractions, long like holding a contraction of the muscle group with the injured tendon for like 30 to 40 seconds or more. This has been shown to increase collagen synthesis, primarily through increasing blood flow to the tendon. Now you might be a little confused there. You might think, Dr. Miller, you just told me tendons don't have much blood supply. So why would doing an isometric like increase blood flow to something that doesn't have much blood flow? And I don't particularly know the reason for this, but the reason that tendons have poor blood supply, it's not often because they lack like the vascular connections. Like for cartilage, cartilage literally doesn't have many, if any, vascular connections. Tendon is a little bit different. Tendon often does have some vascular connections and often substantial amounts of vascular connections, like directly going to the tendon. But for whatever reason, under normal circumstances, the body doesn't send much blood to it, even though it's got those connections. Okay, performing those long, sustained isometric contractions has been found to be a trigger to get the body to realize, oh, you're doing this for a while. You want me to send some blood there. Okay, or you're doing this for a while. Maybe I'll develop some more connections there to be able to send more blood there. Okay, so that's a popular thing these days in injury recovery for tendons in particular in physical therapy. These long isometric contractions, which have been shown to increase blood flow to injured tissues like tendon, get some extra nutrition in there to repair faster and more effectively from that damage. Okay, all that was about tendon. What about ligaments? Tendons and ligaments, of course, are quite different at the tissue level. Ligaments connect one bone to another bone, so a ligament originates on a bone and inserts on another bone. Tendons originate on muscle fibers and insert on a bone. So at the kind of macroscopic anatomical level, tendons and ligaments are quite different. At the cellular/molecular level, at the microscale, tendons and ligaments are quite similar. You'd be hard pressed to look at tendon and ligament in an electron microscope and tell which one is which. They're very, very similar structurally and functionally at the microscale. So all that business on the uncrimping of the collagen fibers and the untwisting of the peptide chains, that all happens in ligaments just as much as it happens in tendons. So if you look at a force extension relationship for a ligament, you'll see kind of that same, two-phase nonlinear relationship that you see for most tendons, where it's got this initial low stiffness stage and then this higher stiffness stage as you ramp it up from moderate to high forces through those same mechanisms that we talked about for a tendon. Now, ligaments have this interesting concept that's been referred to as cellular heterogeneity, where ligaments, just like tendons, have cells in them. They are responsible for the same thing that the cells and tendons are responsible for in terms of engaging in the metabolism and regulating the metabolism that sustains the strength of the extracellular matrix and tears down old collagen and replaces it with new, hopefully stronger collagen. The cells and ligaments do all that same stuff as they do in tendons. Some ligaments are much better at recovering from injury than other ligaments. The classic example is the MCL versus the ACL. Having like a moderate like strain injury of the MCL is of course a pretty serious injury, but it's one that many athletes recover from fairly quickly and fairly effectively. So like if your favorite athlete on your favorite basketball team undergoes like an MCL strain injury early in the season, that's not, there's of course different, grades and severities of all these types of injuries. So I shouldn't speak in generalities here. But if they undergo a moderate strain injury to the MCL like early in the season, odds are, especially with like Division 1 like rehab and athletic training and physical therapy resources, odds are they're going to be able to recover fairly well and probably come back at pretty close to full strength without missing like a massive part of that season. So the MCL is fairly good at healing fairly quickly and fairly effectively from like moderate strain injuries. The ACL, on the other hand, is not so good at recovering from those types of strain injuries. So you see this cellular heterogeneity in those ligaments in terms of some of them having more of these cells that engage in the metabolism than others that could potentially explain that disparity and that ability to repair and recover from injuries. And you also see this in some tendons. So, for example, here is a study examining this cellular heterogeneity. This was in mice, but we might expect to see perhaps a similar thing in humans and their MCLs and their ACLs, where we see this cellular level heterogeneity in the concentrations of different cells in the ACL versus the MCL. So this what's shown here is far beyond my limited capacity of understanding of cellular level biochemistry, but the red dots are indicating a high concentration of cells, and the gray dots are indicating a not so high concentration of the cells. And you can see here in the MCL, we see a lot more red dots than we do in the ACL, indicating a higher concentration of those ligament cells in the MCL, which is a tissue that's fairly good at recovering from injury, versus the ACL, which is not so good at recovering from injury. Okay, that is all for today.